the first stage of meiosis. In this stage, the chromosomes begin to condense and they also pair up. Each chromosome aligns with its homologue partner so that the two match up and begin crossing over in order to exchange DNA. This leads to formation of new chromosomes with unique combinations of alleles. The next phase is metaphase. After crossing over, the spindle begins to take the homologue pairs and move them towards the center/middle of the cell, or can be referred to as the metaphase plate. At this point, each chromosome attaches to microtubules from just one pole of the spindle, and the two homologue pairs of a pair bind to microtubules from opposite poles. Now we are in anaphase 1. In this stage, the homologues, which are currently in the middle of the cell, are pulled apart and moved to opposite sides of the cell. However, the sister chromatids of each chromosome remain attached to one another at all times and don't come apart. In this stage, non-disjunction could occur, such as trisomy 21. This occurs when the homologous chromosomes move to the same side, causing an individual to have 3 copies of a chromosome. This is when the chromosomes arrive at opposite sides of the cell. The chromosomes are enclosed in nuclei. Then the cell now undergoes a process called cytokinesis. This is when the cytoplasm of the original cell divides into 2 daughter cells. Each daughter cell is a haploid and has only one set of chromosomes, or half the total number of chromosomes of the original cell. Now we're on to meiosis stage 2. You can think of meiosis 2 as mitosis for haploid cells. The cells that enter meiosis 2 are the same ones made meiosis 1. In prophase 2, the chromosomes condense and the nuclear envelope breaks down, releasing them to move freely. Then a spindle forms between them, and the spindle microtubules begin to grab chromosomes. The 2 sister chromatids of each chromosome are captured by microtubules from opposite spindle poles. Once again, the chromosomes line up along the middle of the cell. The sister chromatids separate and are pulled to opposite poles of the cell. One of the final steps, the nuclear membrane forms around each set of chromosomes and the chromosomes decondense. Finally, cytokinesis is when the cell splits the chromosome sets into 2 new cells, forming the final products of meiosis, which is 4 haploid cells, which each chromosome has just 1 chromatid, and the final results are gametes, which are sperm and egg cells. And unlike in mitosis, these cells are not identical, they are different genetically.